controlled test. That is the end of term 1 exam or test for life science grade 12. It is M.Saidi. We have picked one of the papers, which is Eastern Cape for 2025. So let's use it as a revision as we are making the memo of this paper. Some answers might be a little bit different because of the variety of different answers which can be applicable to some questions, meaning that your answer might be linked to a question which has more answers. So I'm not going to discuss each and every answer. So I'm only going to discuss one or two answers if those options are there. All right, M.Saidi as usual. Let's start without wasting time. It was the 50 marks, 1 hour. The first question is saying that the region where two chromatids meet together. When you look at a chromatid, we are looking at if this is a chromosome. If this is a chromosome, yes, this point is what they're asking for, point A. So the answer is centromere. So that is 2 marks. They are saying that if 10% of the bases in the molecule of DNA are adenine, what is the ratio of adenine to guanine? Adenine to guanine, that's what they are looking for. So if I have adenine is equal to 10%, automatically we know that adenine goes with thymine. If this is adenine is 10%, automatically even thymine is going to be 10%. So the remaining, if I add the 2, 10 plus 10, it gives me 20. So it means that the balance from 100 should be cytosine and guanine. Therefore, I will say that 100 minus 20, which gives me 80, divided by 2. So I get 40. So why do I divide by 2? Because there are two bases of each is equal to one another. So it means that each is 40 here and also here is 40. So it means that if I look at adenine, adenine is 10 and then guanine is 40. So which gives me divide by 10 so that I get the simplest formula or a simplest ratio. Divide by 10. So it gives me 1 to 4. So you see it's starting is 4 to 1. This is wrong because the answer is supposed to be adenine to guanine, not guanine to adenine. So I have to look for 1 to 4, which is D. So D becomes the most correct answer. They're saying that a woman, found stabbed to death in a hotel. Forensic scientists took a blood sample from the 3 suspects, compiled DNA profile. The DNA profile were also compiled from the victim's blood and skin tissue found in the victim's hands. The following diagram show the DNA profile of the skin tissue sample and the blood from the victim and the 3 suspects. All right. So here they gave what you call the errata. I think just to make it, yes, it's like this. The errata looks like that. So if you look at that, what's the question asking? They're saying which one of the following or which suspect is identified as a murderer. All right, we come back here and then we look at this. This is the skin. The skin, and then this is the victim. So it means that anything which resembles the victim, so it's ruled out. So you have to compare skin and the suspect samples. So this goes with this, nothing here, nothing here. This goes with this, nothing here, nothing here. This goes with this, and then I think it's this, I think so. This goes with this, and there's nothing here. Then we continue, nothing here. So we see that this one is the one which seems to be, so that goes with that, this. You see that, yeah. So basically suspect 1 is the most correct answer. So when we come back here, we look at A as the most correct answer. So we get 2 marks there. We got the next question. They're saying that this is the menstrual cycle, the graph. The diagram below shows or represent the ovarian cycle and the hormone that is controlling it. So we're saying that we see here, we know that this is day 14, that's ovulation, and the hormone we expect here to be luteinizing hormone, which is increasing highly on day 14. Which one of the following combination is correct event that occur on day 14? And the hormone ovulation perfect is wrong. Not. Ovulation perfect, and then is luteinizing hormone. So we see that we talked about it. So D becomes the most correct answer. Yes, guys, I think those people who wrote this paper, I think you see that you're able to get the marks. It was not that a difficult paper so far. And then if you got 8 out of 8, you clap your hands, clap the hands for yourself. And then we can see that, let's go to the terminologies, or what you call the biological terms. So you're saying that a reproductive strategy where young ones are receive nutrients through placenta. When we talk about this, we are talking about vivipary. Yes, so it is a viviparous condition. Then you're saying that extraembryonic membrane, remember extraembryonic membranes, you have allantois, chorion, yolk sac, and then amnion. So it's extraembryonic membrane that is responsible for excretion of nitrogenous waste, or excretion of waste in the egg is allantois. Yes. A structure in a female reproductive structure that carries the fetus during pregnancy. Remember, fertilization occurs, then the zygote will move downwards, and then once it reaches downwards, it starts to form that ball of cells as it is moving downwards. So implantation occurs in the endometrium. But remember, endometrium is part of uterus. And then the uterine wall is also part of uterus. The space in between the different parts of the sides of the endometrium is also part of the uterus, which we call the uterine cavity. So in actual sense, that structure, we can call it uterus. However, some people, they can call it a womb, but scientifically is a uterus. All right. Development where offsprings are born able, unable. Oh, so they cannot feed themselves and they cannot move. So in this case, we shall say that it all depends on the parents. Therefore, we shall say it is artificial. Yes, artificial. Yes, artificial development. So that is the most correct answer. Then we go to the next question. They're saying that is both A and B. Or yes, here you have to be specific on what they're telling you to do. Don't just say A, B, both A and don't say A and B. No, you have to say A only, B only, both A and B, or none. If your teacher gives it to you, if you have written it as A, that is her or his, depending on his mercy, okay, or his or her mercy, but it's wrong. It's wrong, and in final exams, you're most likely to get it wrong. So make sure that you do it in the right way, following the instruction. So we are seeing that the results of variation is crossing over, correct. Random arrangement of chromosomes, yes, during metaphase, both are correct. So I will say that the answer here is both A and B. Then they're saying the discovery of double helical structure of DNA. Rosalind Franklin was among the people who discovered the structure of DNA, who were involved in the discovery of structure of DNA, but he did not find out that is double helical structure. So the only people who discovered this is James Watson and Francis Crick. So this one becomes wrong. And then Gold Mendel is in genetics. So this one also becomes wrong. So the answer here becomes none. Then we go to the next question. That is section A. Section B. So you guys, if you got 16 out of 16 in section A, thank you so much. You are you are still having 100% of your final exams. I'm talking about at the end of the year. So we go to the next question. I told you that once you are given such structure, make sure that you are able to answer all what is being given. So this is DNA structure, and then this process is basically transcription, and then this is the ribosome, and then these are amino acids joined together to form a polypeptide chain. All right, let's start. Identify molecule P, molecule P, molecule P. We say that is DNA molecule, DNA molecule, molecule. Yes, it's DNA molecule. Organelle Q is ribosome, ribosome. Yes, and nitrogenous base R on messenger RNA. Nitrogenous base R, if I'm counting it from the first one, then it means it's uracil, uracil. Uracil. So they want nitrogenous base R is uracil. Anticodon S. Anticodon S. Unfortunately, I might not know what is S here. I might not know what is S here. Yes, if this is the S, if this is the S, yes. So it means that I will go back here. Remember, this is messenger, but they said anticodon, which means on the transfer RNA. So I will say that it's gonna be GGU. Yes, if I take that route. But based on what I see, if this is the first amino acid, it means that was the first amino acid. This is the second amino acid, the second amino acid, and then now this is bringing the third amino acid. So it means that this thing is going to have to move and then to be here, which means that they are talking about the last amino acid, of which is going to be UCU. You see, so the answer is that. So so this question was supposed to be either they were supposed to be specific and then they describe it anticodon S. Here they just say S. We don't know where the S is. Where the S is here or is here or is here. Where is the S? Because they're saying anticodon S. If this is the S, so the question was supposed to be, this S was supposed to be shifted either that side or this side so that you're able to identify which anticodon is it. But based on this, if we are looking at if S is put here, yes, on the second one, therefore this is going to be the answer. But if you follow how many amino acids they have brought, this the first amino acid goes with this, the second amino acid goes with this, meaning that the third amino acid, which is now being brought to be attached to the chain, which is the third one. That's why I read this, and then this becomes the most correct answer. So your answer could either be this or that. I think I've just given it like that because the question here is not that clear. So the examiner was supposed to put that S in the right position, not just hanging to confuse the kids. So, but now you know how to use it to obtain how to, if you are being asked that question, for those people who are preparing, not those who have written this paper. Yes. So they're saying that name and describe the process of T that occur in the nucleus to produce the messenger RNA. So automatically that is going to be transcription. So in this case, you mentioned transcription is 1 mark. Now you have to look for other 4 marks. So you describe transcription. You can use the distinction material book. This one I don't waste too much time because the answer is straight. So you can explain it. DNA double helix unwinds, weak hydrogen bond break. Then it unzip is the same as weak hydrogen bonds break to form 2 separate strands. One is acting as a template. You see it here. This is the one which is acting as a template for formation of messenger RNA using free floating DNA, using free floating messenger RNA nucleotide, or using free floating RNA nucleotide from the nucleoplasm. a complementary strand of messenger is formed with the coded information. Yes, remember it uses whereby complementary strand whereby adenine goes with uracil and the guanine goes with cytosine, meaning that a complementary strand with the coded information from DNA is formed, which is a messenger RNA. It moves out of the nucleus via the nuclear pore to the ribosome. So we are just looking at 4 marks. There we have to look for 4 marks, meaning that you have to talk about 4 facts in a sequence. In a sequence, it must be in a sequence so that it brings about the meaning. Yes, and then you are able to get those 4 marks from there. So the paper was not that difficult. So it means that you have 9 marks there. If you have got 9 marks in this question, thank you so much. Congratulations for that. Oh, the question is still going. The diagram below shows the sequence of nitrogenous bases strand which code for a particular protein molecule. They're saying that now you need to know something here. If they're saying that, what is this? It's a DNA molecule. So if it's a DNA molecule, this is a DNA. What is being asked here is they're asking about messenger. Yes. Write down the messenger. So if they are writing down the messenger, so I'm going to have to come here and say G goes with C, A, T goes with A. So here you're going to say U, A, C, and then here you're going to say A, C, C. So basically that is what you are supposed to write, meaning that C, A, and then U, A, C, and then A, C, C. So yeah. And then you get your 2 marks, meaning that you're supposed to get, it could be all or nothing law. You get it all of them correct. If you fail one, you lose all. It will depend on the final discussion of the memorandum. Then they're saying that valine, okay, the following diagram shows the anticodons of 9 different transferrin A molecules, each carrying a particular amino acid. 9 different, okay, select and write down from the diagram above the correct sequence. They want the correct sequence that would be required for the base sequence of messenger RNA shown above or below. Okay, you have this. This is messenger. But what is here? What is this? What is this? The diagram below shows the transferrin A. So since it's a messenger, then automatically I have to convert it this first. So A goes with U, C, then A. And then it's gonna be C, A, and then it's gonna be UAG, UAC. So UCA, I'll come back here and I'll look for UCA. So the first amino acid is gonna be serine. It's gonna be serine. So your answer here was supposed to be serine as the first answer. Then CAA is tryptophan. Is tryptophan. Is it tryptophan? CAA. Not SCC. Eyeball. CAA is valine. CAA is valine. So the next answer is what? Is valine. And then UAC, 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 UAC. Wait, UAC. USC. USC. I'm seeing USC as methionine. I'm seeing UAC is methionine. Is what? Is methionine. Yeah, so it means that the last one is methionine. Methionine. Yes. So your answer was supposed to be like that. Since these 3 marks, maybe each you write in the correct sequence, then they give you 1 mark. think that is it. Then they are saying that explain how different protein would be affected if the first codon in the sequence changes from this to this. So now, once they give you this question, you have the mutation is always gonna come. You want it or not, it will come. So they're saying that if you do like this, you have to change this, which is U, C, A, and then this one is gonna be U, U, A. So I need to know what is this and what is that. So I need to know that. So U, A, C, U, U, C, A, U, C, A is serine. Here showing it's what? Serine. And then UUA is where is it? Oh, is UUA is asparagine. Yes, it's asparagine. So it means that this is the answer. So it means that when you are writing, you're gonna explain that. What changed here? I think it's the second. Yeah. Since these 2 marks, I think they will mark this, but in most cases, you explain it like this. The second base changed from G to A. You understand? Which resulted in UCA instead of, which resulted in UAA instead of UCA. And a different amino acid is formed. That is serine. No. That is aspartate, asparagine, instead of serine. Yes, meaning that it means that in this case you're gonna tell us that a different amino acid will be coded for. Yes, this one instead of this one, leading to a different protein. But in most cases, sometimes it comes 4 marks or 3 marks. So if they ask you 3 marks, sometimes you tell us that it changed this, since they talked about it. Yes, this will code, and in this will code for this instead of this. You understand? Resulting in a different amino acid, which is this instead of this, hence a different protein. You have to mention them. You have to mention what, you have to tell us what changed and which amino acid formed with it. Is it, if it's different, then it means that it's a different protein. If it's the same, then it means that the protein will not change, however much a mutation occurred. Why? Because the code or the anticodon code for the same amino acid. Yes. For example, if this was serine and this one was serine, then automatically we would have said that there will be no change in a protein. Why? Because it's the same amino acid which is being coded for. Do you understand? But now there is a difference in the amino acid, therefore the protein will change. Yes. So please go to the exam when you know how to use this. And also, please go to the exam when you know this. So guys, apart from this, those people are going to write, please make sure that at least you know that it's the structure of DNA, then DNA DNA replication, the process of DNA replication. Yes, apart from what we have discussed now. And then you can also talk about what's the difference between the nucleotides of DNA and nucleotides of RNA. Yeah, it must be linked to that so that you're able to just practice many questions. You will pass 100%. Well, guys, who got 16 out of 16 this section? If you got 16 out of 16, congratulations for your practice. All right, let's go to the next question. What is A? A, if you look at it, if you are looking at the whole of the structure, it is a chromosome. But if you look at only one side, it is a chromatid. But in this case, we see it's a chromosome. So what is B? It is a spindle fibers. And then, okay, this looks to be prophase 1, this looks to be metaphase 1, and then this looks to be anaphase 1. Why? Because of the chromosomes are homologous. See, they're still homologous. Yes. Then number 2, there is crossing over taking place here. Yeah. So identify what is part A. A, we have seen it. It said that it is chromosome. What is B? B, we have said spindle fibers. Diagram X, Y, and Z are not placed in order. So the answer is Z. You start with the prophase, then you go to metaphase, and then anaphase. So it's gonna be Z, Y, Z, Y, X. It's just a reverse. Then they're saying that use the diagrams, give 2 reasons why the phase are not are part of meiosis 1. Why do you say it's part of meiosis 1? First of all, we see that there is crossing over which is taking place here. That's number 1. Number 2, chromosomes are homologous. Look at metaphase 1. They're still in homologous pairs. They're still in homologous pairs. Therefore, we shall say that this diagram is funny. Ne? How can crossing over takes place? Check this. How can crossing over takes place during metaphase? No, man. All right. That's it. That's how they didn't ask about it. So I was just trying to show you some of the errors which could be found in some papers, yes. But the people who said, thank you so much. At least you gave us what we can discuss today. All right. They're saying that use the diagram. Okay. State 2 reasons why meiosis is important to the living organisms. First of all, it results in haploid gametes. Number 2, it maintains the chromosome number from generation to generation. Number 3, you can talk about variation. Yes, so it brings about genetic variation. So 3 of them, but they only want 2. I think if you wrote that, you are able to get the answers. Then we go to the next question, which is question 3.2. So it's just an extract whereby you're supposed to look at it and then you answer the questions in the extract. Okay, sea urchins release their gametes into the environment, which may be as small as the tide pool or as large as the ocean. The high number of adult and large quantities of sperms and eggs that are produced by the individual sea, okay, urchins is one of the strategies to increase the chances of the gametes making contact with each other. And then in okay, column, okay, many species have further evolved in mechanisms where the eggs release a chemical that attracts the sperms towards the eggs. So it's like that. Females, they have to attract. Yeah, all right, let's continue. I won't say it. This makes the sperms to be the only attracted to one egg of its own species, meaning that other species, yeah. I wonder if you are being attracted to a different species. For example, a dog, a cow. Yeah, that's what I'm saying. Is it possible? So this is just to make sure that the attraction only happens between the same species. So once the zygote is formed, it develops into a swimming larvae or larva. If it's one larvae with the E, if there are many that feed the algae. All right. Identify whether the sea urchins is fertilization is internal or external. Obviously, it is happening outside the female's body. So the answer is external. Why is the sea urchin's reproductive strategy not considered to be ovoviviparous? Is because the development of the embryo is not happening inside the female's body. Yes, it's not inside the female's body. It is outside. Yes. State 1 advantage of sea urchins larvae undergoing precocial development. What is how is this important to the sea urchin's larva to be precocial? So basically, first of all, you can talk about that it reduces the parental care. It reduces, so meaning that the parents won't be always be there looking for the food and stuff. Yes, that's number one. Number two, it increases the survival rate in the high predation environment. You remember, these are too much predation in this water. There's a lot of organisms which will need to feed on them. But since they're able to move immediately or as larvae, then it means that it increases the chances of these larvae to survive in a high predation environment. Yes. So it depends on which answer you give, because this is a broad question whereby one or two or three answers can be applicable. Yes, I think that is it. If you got 4 out of 4, congratulations for that. Let's go to the last question. That is question number 4. Not for question 4. Question 3.3. Scientists think that router graveolens, whatever, L, chemical substance extracted from the plants may be used to the male as contraceptive. Oh, all right. I'm hearing they conducted an investigation to determine this substance decrease the sperm mobility, meaning that it decreases the sperm speed, the speed at which the sperms are moving. Investigation was conducted as follow: 42 male albino rats. Oh, they did it in rats, not in humans. All right. Were used for the investigation. The rats were divided in 6 groups of 7 rats each. The rats were all fed with food, given enough water, and kept at the standard condition of temperature and humidity. All right. They're saying the first group served as a control group, was only given water only, okay, orally, prepared this substance, plant extract 5 grams per kg, was administered orally to other 5 groups of male rats. The sperm mortality, what's the difference between mortality and morbidity? Remember, if you add there R, then it means something else. English. Mortality means that you are ability to walk independently, while mobility is the ability to walk freely. Huh? Is that different? Your ability to walk freely, ne? But mortality means the ability to walk independently. They sound the same, look the same, whatever. Let's continue. We are not in English. Okay, was checked under a microscope under sorry, after half an hour, 2 hours and 4 hours. After half an hour, that's the meaning, 1 hour, 2 hours and 4 hours. 6 hours later, this was done by counting the ratio of immobile sperms, you see. Not immotile, whatever. Immobile sperms to mobile sperm cells per 200 sperm count. Meaning that the graph below shows the mobility of sperms. Okay, mobility of the sperm percentage of albino rats over 6-hour period. Too much reading. All right. The percentage of immobile sperm rate over a period of 6 months. Percentage of immobile sperm. Percentage of immobile. Meaning that what we see here, these are sperms which cannot move. You understand? Yes. Okay. Name one gland in the male reproductive system that secrete fluid in assisting in assisting the sperm mobility. Remember, we said that this was a little bit changed. So they're saying just one gland. So we can talk about Cowpass gland. Cowpass gland. Yes. What about those who talked about prostate gland, and then those who talked about seminal vesicle? Yes, it could be correct because of the generalization of the question now of this answer recently. Remember, I raised the video which explains how did they change it? How they change this? Yes. Credit goes to Mem Maharaja Vadika from, is it Gauteng? I think it's Gauteng West. Thank you so much. All right. Calculate the percentage decrease in the mobility in the time interval of 0.5 to calculate the percentage decrease in the mobility in the time interval of 0.5 to 1 from here to here. So remember, percentage decrease is could be, here they're saying, hey, wait, they're not asking for percentage decrease in actual sense. Calculate the percentage decrease in the sperm mobility. Decrease in sperm mobility. Remember, what we have here is immobility. Do you understand? So it means that they are looking for the percentage increase in the immobility, because they're saying percentage decrease in the mobility. Hey, yeah, examiner. Yeah. Anyway, we use the same formula. Even if you use the other formula, still you're gonna get the same answer. Yes. All right, let's look at the formula we usually use. It's gonna be final minus initial over initial times 100. So we're gonna have to find the final. So the final looks to be, is this 765? It doesn't look 65. Looks 64 minus initial, which is here because we started here up to here. Initial, it looks to be 45. 45 divided by the initial, which is 45. So when I calculate, I'll be able to get, let me check, is 19 over 45, still times 100, which gives me 42%, 42.2 or 42%. What is this? Is the percentage decrease in the mobility of the sperms. You see, percentage decrease in the mobility. Percentage decrease. Decrease in the mobility. Hey, percentage decrease in the mobility, meaning that it decreased. Remember, this is immobility. Ne? At least here, 45% were not able to move. But I still have the 55% they're able to move. Are you seeing? But at this hour, yes, what happens here? How many? I have 65% they're not moving. Only 35% they're able to move. I think. So there is a decrease. There is a decrease in the mobility. There is a decrease in the mobility of the what? Of the sperms. And then in actual sense, you are saying that the percentage of immobility increases. You see? Yeah, this question, I like it. All right, yeah. But in actual sense, there is no science here behind it. It's just that guys always know that final minus initial divided by initial times 100. That's it. If you get a negative answer, it indicates a percentage decrease. Yes. Why didn't we get a negative answer here? Here is because in actual sense, they're asking for percentage increase. You see that it increased the immobility, the ability, the failure of the sperms to move increased. That's the meaning. Yes. The immobility, the percent. However much here, they said it decreased, but the question here is a negative. So that's why you see that there is no negative on it. Yes. All right. After how many hours was the plant extract most effective in decreasing the sperm mobility? After how many hours was most effective? After how many hours? After 1 hour, because here is the most effective. After that, now the sperms, they started to be more mobile. You see, more tired. Yes, so it means that here is 1 hour. After 1 hour, explain why plant extract, this one cannot be considered as ineffective in contraceptive. There is still another percentage. Imagine this, even if we take for the most effective, it's still 35% of sperms that are still motile. They can make you to inject the ovum and then automatically become pregnant. Yes, okay, guys, don't forget to subscribe and like, share to others so that they're able also to benefit. He has been M.Saidi. We always having classes here. Please make sure that you subscribe. Like those who want private classes, contact us. We will be able to provide those private classes at a very small fee. Yeah, I think that's it. Those people who are going for the camp, yeah, we are ready for March camp and also September camp. Yeah, we invite everyone who is willing to come and join us. We will teach you to see that you are able to pass your exams. Thank you so much. I will see you again in the next class.